Hello. Today we're going to take a look at the history of genome mapping. So, what is a genome? A genome is like a map of our DNA. Humans, for example, have over 3 million base pairs. DNA is organized into long strands called chromosomes that exist in the nuclei of our cells. Genes are specific strands of DNA that specify traits. For example, in humans, this could be blood type or eye color. So to go back in time a little bit, who pioneered genes and traits and the research associated with them as we know it today? Well, in the 19th century, Gregor Mendel was the first to associate the relationship of passing traits from parents to their offspring and a relationship across those traits. He studied a variety of organisms, but most famously pea plants, and found that certain traits, such as the flower color or whether the peas were smooth or wrinkly in texture, were passed down from generation to generation in what was most importantly noted predictable patterns. This led to the discovery of concepts of alleles and the idea that genotype, the types of genes we have, determine our phenotype, or the physical trait that is displayed outwardly. Many traits have dominant and recessive alleles, meaning that while a parent or a child may carry one dominant and one recessive allele, they will likely present with the dominant phenotype. However, these alleles can then get passed down to other generations and their offspring, leading to different gene expression. So let's get into genome mapping. Where did that history begin? Well, Thomas Hunt Morgan, using some of the work that Gregor Mendel came up with, he translated that into studying fruit flies. And what he found was that chromosomes that are passed down from parents can experience crossover with their offspring, meaning that parts of genes in different chromosomes can cross when the mother and father genes mix to create a new gene expression in the offspring. Morgan had a student, Alfred Sturtevant, that inferred crossover could be a way to estimate the difference across genes in a genome. And this became the foundation for genome mapping. In the early 1900s, Alfred and Thomas were able to make the first map of the fruit fly genome. So this brings us to the Human Genome Project. What was the Human Genome Project? It was an international research project where the goal was to map the entire human genome, which consists of over 3 billion base pairs and more than 20,000 different genes. It took 13 years to complete this project, and in 2003, they were able to wrap up. The research was able to identify where genes existed on the genome. However, we're still in the process of figuring out what each gene does in the human genome. So work is ongoing there. However, the Human Genome Project has laid the foundation for biologists and genetic work to continue worldwide. So there's still a lot to uncover in the exciting field of genetics and biology, but mapping genomes has been an incredible feat that we've accomplished. That concludes our presentation on genome mapping. Thank you.